Okay, this is the last of the lessons. So covers mutations and sequencing. I think that's pretty well it. So um, I'll try not to get all teary-eyed. Let's go. Um, we've talked about mutations before. This is going to talk a little bit more about some other types of mutations. So mutation, like we saw before, is a permanent change in the genetic material of an organism. It's positive in that it can allow for genetic variation. Oops, sorry, I'll get the pen out. So sometimes mutations can be a positive thing, which can allow for natural selection. Sometimes they may lead to negative things like diseases or disorders. Some may be harmless. There are spontaneous mutations and there are some that are induced. So spontaneous simply means that it arises as a result of molecular interactions in the cell. It just kind of seems to happen. Nothing really is causing it to happen. So it happens spontaneously, usually during one of the three processes that we talked about. So either DNA replication, during the transcription process where it's being converted into a message form, messenger RNA, or in translation, that there's errors in the amino acids that are put together. Induced kinds are those that are caused by some kind of an agent or event outside the cell. It's called mutagens. Mutagens may be physical, like UV, or they may be chemical, like carcinogens. Okay, so we have things like viruses or bacteria or things like that that can cause mutations to arise as well. So we don't have to know a whole lot about them. Okay. Chromosomal mutations are ones that there's a mistake that may involve more than one gene. So we talked about these in the genetics chapter, where it could be an inversion, translocation, duplication, or deletion of whole sections. So chromosomal mutation involves more than just maybe a certain nucleotide. It is like a gene or more than one gene. A gene mutation is a mistake in that single gene, so an incorrect sequence or message. So it could be a deletion, an insertion, or a substitution of a base pair. So chromosomal involves more than one gene, like a section of a chromosome, whereas a gene mutation is within a gene itself. Okay, if it's, we're going to look at pictures of these too, but if it's a deletion or an insertion, then it causes a shift. It's called a frame shift mutation, where it shifts the codons. Okay, so if you look at the example that's given, this is what makes sense. Okay, this is the normal codons. The fat cat ate the big bee. But if there's a frame shift, then it's going to shift the codons either one to the left or one to the right, and then it doesn't make sense. A germ mutation is one that happens in the sperm or egg, the reproductive cells. that means if it happens in those, then every cell of the individual is going to have that mutation. If it's a somatic mutation, it occurs in the rest of the cells, and it's probably just going to be localized to that region. So if, say it's caused by UV, and the UV was focused on the section on the back of your shoulder, then it's not going to affect all the other cells in the body. It's just going to affect those ones in that region on the shoulder. Those would be the kind that end up causing like cancer, melanomas, things like that. Okay, those are kind of terms and definitions. So let's have a look. These are the ones that you learned about in the genetics chapter. So it involves, these are chromosomal, it's a section of a chromosome or involves more than one gene. Okay, so these ones you will have to know for the exam. Probably not quite as involved for this chapter, but it's to understand the difference between them. Okay, so inversion, where you have a section that kind of has come out and reinserted itself the opposite way. Translocation, where you have a section of one chromosome being translocated to a section on another chromosome, not a homologous one. Or it could be a homologous one, but normally it's to another chromosome. Deletion mutation, where a section of the chromosome is lost. Duplication, where a section of the chromosome is repeated. Okay. Gene mutations, on the other hand, if you look, this one would be an example of a silent mutation. So here's where the mutation happens. ACU becomes ACC. But if you look on your chart for codons, both of those codons code for the same amino acid. So since they code for the same amino acid, it does not affect the protein at all. It is a silent mutation. Okay, so the silent one wasn't listed on there, but it is another kind of gene mutation. It just doesn't really have any effect on the protein or its job. Okay, so it is a type of substitution that is considered silent because it doesn't affect the protein, doesn't affect the amino acid or the protein. Here's another substitution. Okay, so normally it's GAA, but this substitution becomes GUA. So the rest of the protein would have the right amino acid sequence, but this one changes from glutamate to valine or valine. Okay, so depending on where that sits in the protein, it may alter its function a little bit, it may not alter it much, or it may make it so it's not effective at all. So that one is a regular substitution, not a silent one. Okay. Let's see. This one, if you look, it's normally CAUUUG. Here, there is an extra G put in right here. Okay, so you'd have GUUGAC, UUUGAC is here. Okay, so because of the insertion, because it's an insertion, it changes all of these amino acids. It causes a frame shift and changes them. Okay, if we look at the last one, the A here is lost. So because the A is lost, it's a frame shift mutation again. This one is a deletion. So from where that happens, all the amino acids have changed, and it may even truncate the protein or might make it so it doesn't get produced at all. So some mutations can be very bad. Some may not cause a lot of change to the protein or its jobs. Okay, the last little section is on DNA profiling, and this is interesting. So I usually include a little section on this, and usually have a question, certainly in the little mini lab, but possibly on the exam where you have to actually do some sequencing. So normally when you're doing a profile, let's say you're trying to figure out who a killer is in a murder investigation, and they find some evidence. The first thing is they have to get some DNA evidence. So it might be from the hair follicles in a brush, or from lip cells from their lips on a cup, or some other source of DNA. So because they only have a little bit, in order to do the studies and repeat the reactions that they have to run, they need to take that DNA and make copies of it. So, oops, I have a pen. I didn't mean that. So they do what's called TCR. That stands for polymerase chain reaction. And you don't necessarily have to know what those letters stand for, but basically what they do is they take that little bit of DNA and they put it into a machine with certain solutions, and that goes through a series of reactions in order to make a whole bunch of copies, kind of like a photocopier. Okay, so it makes a whole bunch of copies of the DNA so that they can send it for analysis. The last step is analysis, and usually that takes one of two forms. One is sequencing, so they actually sequence all of the DNA, and the second is by what's called restriction analysis, and that lets them kind of treat the DNA with certain enzymes that cut it in a distinct pattern, and that distinct pattern would be different. Um, they'd be closer in people that are really closely related, or exactly the same in the same person. Okay, so first thing we're going to look at is the original way they used to sequence DNA, and it's called the Sanger method. In the Sanger method, that's what I used to do whenever I was doing my master's degree. Basically, we had to prepare the DNA, and we added radioactive nucleotides. Okay, so we had a tube where it was C's that were labeled, a tube where it was the G's that were labeled, a tube where it was the A's, and a tube where it was the T's. So every time when we run the reaction, um, it would cause all the different sizes of all the different, oh dear, I think I'll do this one.